Right, I see we've got a lovely audience coming in now, so I'm going to move on and introduce today's speaker, Jane Stewart, Associate Professor at Colorado State University. Jane, I am just passing the screen to you if you'd like to accept the invitation to present. Jane
Thank
is
you
a
so much,
forest
Renee.
pathologist. She is, I'm just going to babble on for a few more minutes, give you time to set up the screen and give you a proper introduction befitting your expertise. As a forest pathologist specialising in emerging pathogens. And Jane is about to tell us about the test that she's been developing for the identification of different Myrtle Rust strains. Jane, you can now feel free to get underway.
Okay, thank you so much. So as Renee mentioned, I'm going to talk about the development of molecular tools for detecting genetically distinct biotypes of Austropuccinia psidii. And this is a collaborative project with myself, Jorge Ibarra Caballero, who is a research associate in my lab, and Drs. Misuk Kim and Ned Kloppenstein from the USDA Forest Service. And as you can see from this title slide, we worked with a lot of different collaborators from Australia, New Zealand, Brazil, and Hawaii to develop these probes that we've been working on. And so without this collaboration, this work would not be possible. So because we're in a Myrtle Rust webinar, I'm not going to talk about the pathosystem specifically as an introduction, but rather I'm going to talk about the overall question that we want to ask here. So our question is, do different strains or isolates of Austropuccinia bacillii that infect different hosts, this is a really broad host pathogen, and from lots of different global areas around the world have different genetic signatures, or they different genetically. And so as you can see on this graph, we basically have Austropuccinia bacillii has been found in the Americas, it's been found in Africa, and it's also been found in Oceania and other areas. So looking at different strains, what's the diversity that we see? So the first person who really started looking at this, or one study that was done, was by Grauke et al. In 2013. And this team basically collected isolates from Brazil and Uruguay and then looked at whether or not there were genetic differences using SSRs or microsatellite markers. So they had over 100 isolates that they collected from primarily eucalyptus and guava. And what they did was they biotyped these, or they genotyped them using microsatellites. So what we have here is a Bayesian structure plot, and it allows us to look at the genetic diversity in the populations across the total group of isolates. And so each bar is a genotype, and the color of the bar represents which population that may have come from. And so what you can see here is that there's basically the best number that they had were 2 different populations, but they're really distinct. So the population in eucalyptus is distinct from the population on guava. And so they also showed, using their data in a PCOA plot, principal component analysis, that there was further evidence for this. So here you can see the guava biotype I've circled in blue, whereas the eucalyptus biotype I've circled in green. And so they're really distinct patterns. So it looked like there are genotypes associated with different host species with Austropuccinia bacillii. The other questions they wanted to ask was whether or not there was a host jump from guava onto eucalyptus, and if somehow these different genotypes were related. So what they did was they produced a minimum spanning network, which allows us to look at how the different genotypes are related to each other. So I'll just sort of describe this first. So each circle represents a genotype. The size of the circle tells us how many individuals have that same genotype. And then the bars look at genetic distance across. So what they did was they color coded the different circles. So black was eucalyptus, and then green was guava. And what we see here is that there's actually quite a few different eucalyptus genotypes, and they're genetically really distinct from guava, right? So there was evidence here that there are separations, but there was no evidence for a host jump, indicating that this was probably something that was introduced into Brazil, potentially. But there was a need to assess whether or not there's different biotypes within Austropuccinia bacillii with a larger global population. So collaborators and we developed this question looking at whether or not there's genetic diversity across the basically North and Central Americas compared to South America. So what we did was we scored 6 microsatellite loci, which were the same ones that Grauke had used in their study. And it was 226 isolates from 28 different hosts and 6 different countries. And here you can see the list. So we had isolates from Brazil, Costa Rica, Jamaica, Mexico, Puerto Rico, Uruguay, and then from Florida and Hawaii in the United States. And then you can see that there's a really broad variation in hosts. So basically what we found when we used those microsatellite loci was 23 unique multi-locus genotypes. And then using a method of a Bayesian structure plot that was developed in BAPS, which is a program you can do similar to what Structure, which is another common program, uses, we found basically 3 genetic clusters in Central and North America, and then 6 different clusters in South America. Interestingly, there was one genetic cluster that actually was found in both areas. So then we also wanted to assess whether or not, how these genotypes were basically connected to each other. And so we produced a minimum spanning network. And again, each of these is a genotype. And what we then did was overlaid genetic cluster onto this. So if we look at this one area here, this red circle, this was one genotype that was represented by 55 different individuals from Hawaii, Costa Rica, Puerto Rico, and Jamaica. And so what we basically found is that there was support for 3 main different biotypes or lineages that basically represent most of the isolates. So here we've got our pandemic biotype, which I've circled in red. And this comprised basically 2 different genetic clusters, the green here, which is C4, and the red, which is C1. But you can see that there's lots of different genotypes within this one biotype that we've designated. We also had another biotype that's called the guava, Brazilian guava biotype. And this is really interesting because this is only comprised of a single genotype that represented 65 different individuals. And then we have a third biotype, C2 and C3, that we designated the eucalypt rose apple biotype from Brazil and Uruguay. And this was represented by 5 different genotypes that belonged into 2 different genetic clusters. We also had some evidence for potentially an allspice Jamaica biotype. You can see the circles labeled in purple here, but we know that we've got at least 3 that are pretty well represented: the pandemic guava biotype, and then the eucalypt rose apple biotype. There's also been data done by Granados et al. Showing that there's one more biotype that has been identified. So they used the same SSR markers that we used in our study as well. And what they were able to show is that here's the pandemic biotype that's similar to ours, but there's actually a South African biotype that seems to be unique from these others. So globally, what we found so far, there's at least 4 different biotypes of Austropuccinia and psidii, the pandemic, eucalyptic, guava, and then the South American. We also wanted to look at whether or not there's differences in the ecology of these different biotypes. So we know that there's probably some differentiation in host specialization, but we also wanted to look at climate space. So this is looking at suitable climate space across the different biotypes. So this is using max entropy modeling. And when we look at all the genotypes together, so this includes the whole population that we have, we basically see that there's suitable climate space in Brazil and in other areas like in Australia and New Zealand. Now, the darker the red means that that area is a more, there's probability of that of isolates being able to survive in that area, so it's more suitable, the darker the red. And then when we start to look at these different panels, so B showing what the climate space would be for the pandemic biotype, C is looking at the eucalypt rose apple biotype, and then D is looking at the guava biotype. And what you can see, if we just focus on Australia here, you can actually see that there starts to be differentiation in the climate space, suitable climate space for these different biotypes. And so it does appear that there is variation ecologically for these different biotypes. So basically what we found in our study, looking at over 200 different individuals, is that in North and Central America, we basically have the pandemic biotype. So it was represented by those 3 different genetic clades. The red and the green indicate the pandemic biotype. And then we also had this other genetic cluster, which was the allspice group as well. But we had less diversity than what has been identified in Brazil. So in Brazil, they actually have more genetic diversity, and there's in Uruguay have been found to have the pandemic, the eucalypt and the guava biotypes. So basically they are geographically distributed as well. So one thing that is worrisome is that we know that these different biotypes are going to behave differently on the landscape. And so there's been, just because you have Austropuccinia psidii in your region doesn't necessarily mean you're not at risk if a different biotype were to be introduced. So there's been some work looking at what that risk might be. So some work that was done by Silva et al. Showed that ohia, which is a really important plant species in Hawaii, that it is susceptible to the eucalypt rose apple biotype from Brazil. So if there were to be an introduction of this eucalypt rose apple biotype, we know that ohia would be susceptible and would likely proliferate. You can see an example of an inoculation there. When we look at our bioclimatic modeling, we also see that there is suitable habitat or climate space for this eucalypt rose apple biotype as well in Hawaii. So there's this risk. There's really a need to try and assess what the risk might be and then avoid getting these new biotypes introduced into different regions. These potential threats have also been looked at through other researchers. So Zhao et al. Looked at what the threat would be if, on Rutaceous species if there was the introduction of eucalyptus into Australia, and there were several different species that are at risk. Our bioclimatic modeling also shows that there is some suitable climate space for this different biotype, for the eucalypt biotype. And then Stewart et al. Recently looked at whether or not native New Zealand Rutaceous species would be susceptible to the African biotype, South African biotype. And what we do find is that there are some species at risk. So really, limiting the spread of these different biotypes is vital to ecosystem survival. So basically from the stuff that we've done so far, we've found that at least 9 different genetic clusters have been identified. We do see that there are 3 different biotypes. So we've got the pandemic, we have the eucalypt rose apple, and then we have the guava. And based on the research that we've done, that we and others have done, have shown that there's differences in host range and there's differences in suitable climate space. So where they move around the world might differ. And so understanding what these differences are, I think, is essential for tracking the pathogen. And just because you get Austropuccinia basidia doesn't mean that those other new introductions might have consequences to the landscape, right? So we need to be able to develop some tools to implement management strategies for this invasive pathogen. So we are working to develop a biotype specific identification tools. The Jorge Ibarra Caballero is a research associate in the lab, and he's the one who's been doing much of the work for developing these marker systems. So the first thing that we've needed for these marker systems are genome assemblies. And luckily, there have been some that have been released, but also we've been working with collaborators to be able to get genome assemblies of each of the different biotypes. So we have assemblies of the pandemic, the South African, the eucalypt, rose apple, and the guava. Luckily, the pandemic has been done really, really well, and it's actually been sequenced, and it's the largest assembled fungal genome to date. The annotation files have included transcriptome, proteome, CDS, and effector profiles, which has really been helpful for designing these marker systems. So basically what you can see is that some of the genomes have been sequenced more thoroughly or as a reference strain than others. And so there's less genetic information for the eucalypt roseapple biotype or genome and for the guava genome, which has sort of changed our ability to develop these marker systems, which we'll talk about. So there's basically two different methods that we could utilize to make these markers. One would be try and find basically novel regions in the genome and develop a marker based on that. But what we found is that this has been really problematic because a lot of these regions have repeats and they're not very good for molecular assays. Over 90% of the genome is covered by transposable elements, which makes it really problematic for marker development. And so the second strategy that Jorge's really been working on is searching for common regions across the 4 different genomes, but that have low similarity across them. And this appears to be a better way to develop these tools. So this is just an example where we've got 2 different contigs here, labeled in red and labeled in green, where this one, you can see that in one copy, there's a hit, but it's fairly low. But then we also have another copy that's really high. So just because you have some contigs that have low identity, you might have another region where it hits again where it's pretty high. And so looking at these across all the different biotypes can be very difficult to discern. Here's another one that's labeled in green where there were low identity hits across the South African compared to the pandemic. And so this would be a region that we would target for marker development. So we're also been thinking about the marker systems that we want to use, right? So in developing molecular tools, you could use something like qPCR or PCR, but you really need a thermocycler for developing these or for using these kind of tools. And what we really want is something that we can do in the field. And so we've been focused on using a LAMP assay which is loop-mediated isothermal amplification. So it's isothermal, so you don't necessarily need the cycling like you would in a thermocycler. And we're able to develop a detection system that's colorimetric. And so it's something that users without any really background experience can use in the field and see a color change of whether or not that pathogen that they find is the one that they're looking for. Now, it's a little bit more complicated to use compared to some other marker systems because there's 8 different primers that you utilize. So it basically, you have a region of DNA, and then you basically need 8 different primers, which are labeled in these colored boxes that allow you to amplify the region that you're looking for. And what this allows is for increased sensitivity and specificity, but it makes it more difficult to work with because you have to find a region of the genome that's going to be suitable for those 8 different primer systems. Now, there is software that you can use. It's called Primer Explorer. And this is really widely used, and you basically put in your region that you're looking for primers for, and then it'll help you design the different primers. And so it's a complicated method, but we've been able to use it successfully for a couple of different pathosystems. And so that's sort of why we lean towards this marker system. So this is showing basically you'd put in all of your primers, right, with your reagents. You'd put in some sample DNA, usually 1 to 2 nanograms, and then you'd get a colorimetric response. So basically, negative reactions are pink, whereas positive reactions turn yellow. And so this is sort of the ideal system that we have. Now, to be able to develop LAMP markers or to test the ones that we've developed, we needed DNA. And so luckily we were able to work with our collaborators to get some DNA. So we've got some from the pandemic, from Hawaii, we've got some from the South African biotype, the eucalyptus biotype, the guava biotype, and then we actually had 2 strains of unknown biotype that we've been working with thus far. So Jorge has developed 2 different color system or 2 different assays for the pandemic biotype. So what we basically have here is the pandemic biotype, or the pandemic assays here, and then we put in different types of DNA. And so basically this 1 is the pandemic biotype, 2 would be the South African, 3 is the eucalypt type, 4 and 5, we don't know what those are. And so they were just sort of test unknowns. And then the guava biotype is in 6. And of course the negative control, which is really important to include. So when we added 2 nanograms of DNA and incubated this assay for 30 minutes, we were able to show that the pandemic biotype did amplify the pandemic DNA, but then it also ended up amplifying DNA from Brazilian guava. And then when we looked at the South African assay, we do see that it amplifies the South African DNA, but it doesn't amplify anything else. So it did look like we do have successful pandemic and South African assays that we've developed. But again, we've got to test these on a much larger scale because we're basically using DNA from one plant sample or one strain sample. And then when we started to look at the eucalyptus assay and the guava assay, under the same conditions, basically 2 nanograms of DNA and 30 minutes of incubation, we see a low sensitivity. So there's actually no positives. So false negatives are arising. When we added 10 nanograms of DNA and then incubated for 50 minutes, basically we do start to see that the eucalyptus assay works, and we do see that the guava assay works. But we've been working to really fine point the sensitivity on these assays, which has been sort of difficult to do because I think of the genomes that we don't have as much information compared to the pandemic and the South African. So we've kind of come up with a strategy where we can develop one assay where the pandemic amplifies. We have another assay that's specific for the South African. And then we've been working on developing an assay that amplifies everything but the guava, and then another one that amplifies all biotypes. And that way, if we can't get one that's really good for guava, we will be able to basically assess if this is positive. We'll know that that one sample is actually the guava biotype. But what we found thus far is that we aren't able yet to develop an assay that works for all biotypes. So we're still testing that and working through these. Another thing that we found is that timing of the LAMP assay is really important. And this is a process where it's time sensitive. So here we've got that same strategy that I just presented. So we've got the pandemic, the South African, the pandemic, South African, and eucalyptus assay, and then the all biotypes assay. And this is after 30 minutes. And what you can see is that the yellow looks more of an orange. And so you get some inconclusive, basically, responses if you're using, if you let this sit for 30 minutes. If we start to increase our time to 45 minutes, you can see that the yellows are really solid and they're real positives, right? At 50 minutes, you see the same thing here where they're good results, really positive. But then when we start to look at 60 minutes, we start to get some false positives where we get some inconsistent results that occur. So timing is of the essence when we're using this LAMP assay. You don't want to run the assay too short, but you don't want to run it for too long either. So what we've been working with so far is getting samples from people, from collaborators. Just recently, we got a sample from Uruguay, from Carla Perez. And what we've been doing first is taking the sample and then identifying them by biotype using 4 different SSR markers that we've used previously in other studies. And what we find is that there's genotypes using those 4 SSRs that are specific to the eucalyptus, the pandemic, and the guava biotypes. So basically what we were able to do is run those microsatellites on that sample. And what we found was that it had basically genotype 2, which makes it fall into the eucalyptus. And then we double checked that using our LAMP assay. And what we see is that using our LAMP assay, it is a eucalyptus biotype. And so when we get samples to ensure that our assays are working, we've got to type them with the SSRs and then run them on the lamps as well. So what we hope to get to is a point where we basically are able to use these lamp markers in the field. And we've developed one right now for Carnosaur rubicola, which is the causal agent of white pine blister rust. And there's a couple of things that we found that work really well. So basically the reagents are stable in -20 for at least a week, and they can stay at a day or so at 4 degrees. We've been able to basically use this assay using spores of rubicola or bark samples, and we extract that DNA in Chelex or in PVP for 30 minutes at 98 degrees. And then we take this solution of the extracted DNA and add it to the lamp reagents. And then we've been able to, we have a heat block that you can actually plug into a 12-volt outlet. And so we're actually able to do this in the field, which is pretty remarkable. And it's something that most folks can utilize. So field crews can utilize this. So there's a couple of caveats that we need to talk about. You can have the reagents change based on pH. And so that color change is really based on pH. You can have false negatives or false positives. And so we're working through the assay and troubleshooting the assay for Myrtle Rust. Also, we notice that leaf tissue alone will cause some inconsistencies to occur. So we basically are scraping off spores off the leaf tissue and adding just spores into for the DNA extraction. And then also we've added this PVP because it helps stabilize the assay quite a bit. We extract the DNA for 30 minutes at 98, and that seems to be important as well. So far, we've really only worked with DNA, and so we're working with collaborators to troubleshoot the application. We have distributed this out to other collaborators in Australia, New Zealand, Hawaii, and they're starting to work on those. And so hopefully soon we'll be able to have more information about how these markers are working. And with that, I'd like to thank all of our collaborators. As you know, this work is not possible without collaboration, so I thank all of you. And then just really one last comment is that this was Malachi in Hawaii in October 2019, and there was a huge defoliation event that occurred. And so what we were able to do is determine, was this weather related or was it strain biotype related? And so we were able to collect samples, and we showed that this was actually the pandemic biotype, which is already there. And so it was likely related, this defoliation event was likely related to weather patterns, not the introduction of a new biotype. So thank you. I'll take any questions if you have any.
Thank you very much, Jane. That was a fantastic talk. And for me, it was great to see your pictures of the little tubes all lined up and the colours. That's not something I've seen before, and it does help one get a sense of how straightforward this could be for people to use. You actually answered one of my questions at the end there around, well, how straightforward is it to actually get the DNA, right? Because you've still got to get that. And I suppose for community groups or even highly interested members of the public, they're probably not going to have a heat block, but certainly something accessible for experts in the field.
Yeah, and the heat blocks aren't very expensive.
Really?
I think they were a couple $100. So
I was going to say,
yeah,
in New Zealand.
Yeah, that's true.
Of course, not all of our viewers will be from New Zealand. Sorry, carry on.
Yeah, but so it is possible to do it. I mean, you have to be near a car. So sometimes field work, you know, you have to get, if you're out in the woods in a backpack, that was a little bit harder. So you do have to get to your car to process them. But you could definitely do it in a hotel room or in your car, for sure.
Certainly could. Now, I've had a couple of questions come through, so I'll just share those with the audience and then I will ask them to you. All right, it's a little long here, so buckle up. What would the recommendation be when using these molecular tools for surveillance and monitoring in areas where the rust is already present? Would we use it routinely in high-risk areas? When we find it in new areas, on new hosts? It would make sense to know what strains are already present as a baseline and their distribution. We don't update distribution regularly enough because of challenges with time and funding. But how good would it be to track movement and spread of the biotypes almost yearly? Well, yes, there's a lot there. I wonder if the main thrust is really just how you prioritize when you've got limited time and money, what you're going to sample and what you're going to target.
Yeah, I mean, it really depends on what the management strategy is for an area, right? So the introduction of a new biotype is probably the most important, I would think, because you might be able to maybe clear cut an area or do some sort of management strategy to stop, like quarantine and then stop the spread of it if you were able to detect it early. But I do agree that monitoring where the distribution is and where these different biotypes are is really important. So you could use it for multiple facets depending on your management strategy for an area.
Yeah, I think that's what it really comes down to, doesn't it? The next question, and of course we have a great talk, Jane, here. Thank you. Is the white pine blister rust lamp being used in the field?
We have started using it in the field, and one reason why Forest Health Protection around us is using it because in high elevation pines, especially in Colorado, it hasn't actually been introduced into the entire area. So quickly identifying is really important. So it is being used by FHP. Folks who don't know how to use a pipette, we teach them how to use a pipette, and they can run the assay themselves, which is fantastic.
Yeah, that is very good. And I guess kind of in line with that question around ease of extraction of DSA, I was thinking about the cost of the whole process, I guess, and how accessible this is going to be to people.
Yeah, we estimate that it costs about $2.50 for an
Wow.
assay, but that's if you already have the equipment, right? So, and it's not that expensive, really. You do need some tubes. You could use a pipette, but they could be one of those plastic pipettes. So I don't think it's cost prohibitive. It's actually one of the cheaper methods to use compared to like digital PCR or real-time PCR.
So we've just got another general thanks here. I will ask a couple more of my own questions in case people are still just shaping their own. And then if we don't hear anything further, we can wrap this up. So I guess my other curiosities were about these different biotypes themselves. And I was wondering, is there much consensus around how much genetic difference is required to declare something a different biotype? And, how quantitative is that?
I think we don't know yet. When we start to look at, well, we had 9 different genetic clusters, right? When we looked at the population in the study that we conducted, and we
So
know
you're
that some
eyeballing
of
them on a
those,
plot to some extent.
yeah, and then basically where they've been found and how prevalent they are on the landscape in the areas where we collected is sort of what we've been going at. I presume that there's going to be more biotypes that are introduced or that are discovered, and so yeah, and if there's sexual recombination happening, which has been shown now, we could get even more diversity mixing. Yeah.
We certainly could, and I guess it just got too tempting here to draw the parallels with COVID when I was thinking about these different biotypes.
Yeah.
We have the classic example here now with Delta versus Omicron, and now we've got Omicron's little sister or little brother or something like that. And obviously, there's a big difference between Delta and Omicron and what's going on. And there's a lesser difference between Omicron and newish sister of Omicron. Do we see the same kinds of parallels, that there's a spectrum of differences between biotypes as well, do you think?
Yeah, I think when we look at the genetic diversity, there are some big steps in between these different biotypes, depending on which ones they are. So yeah, I think obviously the rust is going to evolve slower than the virus would, but I do think that new genotypes are rising, and they seem to have host preferences, which means that they could be pretty genetically distinct.
Yeah, and I guess irritatingly, though, it's likely to be the case that a big genetic difference isn't necessarily going to translate to a big ecological change.
Right.
Nature isn't
I
normally that
agree.
tidy,
Yeah.
is
Yeah,
it?
that's true, right? We've got to keep testing them to see how they vary. And I don't think we even know, like, virulence has been tested, but whether or not they sporulate better, are there differences in climatic regimes? I mean, there are based on the data that we have, but what are those, right? And so, yeah, there's a lot more work to be done to understand what that genetic difference actually, how that correlates to a phenotypic difference in the pathogen.
For sure. Now, we do have another question that's popped up here. It looks like there are multiple lamps required for biosecurity in Australia. Would we just use the pandemic one to start?
Yeah, I think that would be a safe bet. And then I think that's what most people are going to do. So I'm actually been distributing these in Hawaii. So we have a project in Hawaii to look at whether or not different biotypes have actually been introduced. And so that's what they're doing is they're starting with the pandemic. And then if it doesn't amplify or doesn't light up with the pandemic, they're sending the sample to us so that then we can use it to see if it's a different biotype. So that's what is happening right now. Yeah.
Great. So we probably could wrap this up shortly, but I guess perhaps I could ask you to talk a little more on the immediate next steps for this work, and perhaps whether there's anything you'd like anyone in the audience, scientists, collaborators, to assist you with at the moment.
Yeah, we would love samples. So we are working with some collaborators who are using the probes in New Zealand, actually, but we would also like samples sent to us, especially from Brazil, where we've got those different biotypes overlapping. It would be awesome to get samples, real life tissue that we can work with. Right now, we've tested the assay, but primarily on DNA samples rather than spores. And I think there are going to be differences that we see with the different samples that we use. And so to make this as robust as possible, we need a lot of testing so that we can sort of figure out what's the best method. Are these assays actually working well? Are they going to work on all the different genotypes that encompass a single biotype? Yeah, there's more work that needs to be done for sure.
And I guess you don't have perhaps quite the same challenges as we might have in New Zealand with importing plant material, and especially a pathogen.
Yeah, I mean, Hawaii's got a big hurdle, and that I think there are regions where this could get introduced and be really, really, the different biotypes could be problematic. So we have sort of the same issue, but maybe not on the same scale.
And
Geographically,
obviously ways around these things. All right, well, there's no more questions come in, so I think I'll wrap it up there. Thank you very much again, Jane, for that really professional, useful presentation and for sticking around for some questions. A video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and it will also be emailed to everyone who registered. Now, the next webinar will be on Wednesday, March 9th. We will hear from Rebecca Campbell at Plant and Food Research. So she's down here in New Zealand, well known here within the Myrtle Rust community, partly because she is the creator of the map that is currently available on the myrtlerust.org website. It's again a New Zealand website, and it's showing Myrtle Rust records in New Zealand. Now, Rebecca has recently received a fellowship to work further with spatial data and modelling, so that's fantastic news, and she's going to tell us about the creation and maintenance of the map, as well as introduce the new work she has planned. So her talk title, therefore, is Surveillance Database and Mapping plus Spatial Epidemiological Modelling, Learning from Myrtle Rust in New Zealand. So hopefully there'll be some good things in there for our international audience as well. And you can all look out for the invite. That's it for today, everybody. Haere rā. See you next time.
thank you.